Hi guys, Mrs. H here. So this week we're going to be looking at dihybrid crosses in genetics. So what is a dihybrid cross? So a dihybrid cross actually looks at two traits or two genes on two different spots on a chromosome. So sometimes we can use a dihybrid cross when we have a polygenetic trait. So many genes to determine one phenotype. Now, we won't be doing that. We are going to be looking at two phenotypes. So, two genes, two phenotypes. And what are the possibilities that we could see? So, let's look at that. What are we talking about? Two traits. What does that mean? So, here's an example. I have guinea pigs. So, I have a female guinea pig and a male guinea pig, and we're going to look at the coat and texture, the coat color and texture of the guinea pigs. So notice texture and color. And so we can cross those based on texture and color. So that would be two traits, both referencing hair or their coat, but what about their coat? But the color and the texture. So this is just like a monohybrid cross when we're doing 4 steps. So we first identify the parent genotypes. We need to determine the gametes, what we put along the tops and the sides of our boxes. Then we need to fill in our box or our Punnett square and then determine the genotypes and phenotypes. So those 4 steps haven't changed, but they're a little more complicated now because we have two traits. So let's do a cross. Let's do an example. So I have a homozygous tall, homozygous yellow plant, and I'm going to cross it with a short plant that has a green seed. So the first thing I need to do is identify the parent genotype. So for height, here are my different possibilities. I have tall TT, tall T, little t, or short little t, because tall is the dominant trait and short is the recessive trait. And then for color, we have yellow, so yellow is dominant. So homozygous yellow, dominant, big letter Y, big little letter Y. That's going to give us the yellow appearance. And then the green appearance is little y, little y. So again, that hasn't changed. We're now needing to find out what the parent genotype is based on the phenotype. So I have homozygous tall, capital T, little capital Y, little capital T, capital Y, with all lower cases for the short and green. And that's based on using this information up here. So this would be the genotypes of the two parents. So now I need, so again, this homozygous tall gives me the capital T. The homozygous yellow gives me the little y or the big Y. So that's parent number 1. And then the short, little t, and the green, little y. So now I need to, because of independent assortment, these alleles will separate by independent of one another. So I will have a situation where I'm going to produce gametes where each allele can go with the other one. And so what does that mean? Basically, that capital T can go with a capital Y. That would be the first gamete. What else can happen? That second gamete, so the capital T can also go with the second Y. So that would be a second gamete that would be formed. Normally, on a monohybrid cross, you only have 2 gametes formed. But that's not the case in this. We're actually going to have 2 more, because what about that second T? So that second T is going to go with the first Y. So that's gamete number 3. And then again, the second T will also go with the second Y. And so we have 4 gametes that are produced. Now, on this one, it's kind of hard to see because they're all capital T, capital Y, but it's because they've been independently assorted, but they're all the same. So you don't see any difference. So a kind of trick on this is thinking about this as FOIL. The first T with both Ys and the second T with both Ys, inside, outside, right? FOIL, do you hear it? Okay, so the second parent, again, because they're all the same, capital T's are a little case t and little case Y. We didn't really have to do that one because they're all the same. So you're going to put your gametes up at the top of each box, just like you did before. So here's gamete 1, gamete 2, gamete 3, gamete 4. This is why the box is bigger, because we have 4 gametes produced. What's the other one? The other one is going to have 4 gametes produced as well. So I have 4 across and 4 down, with each one of the gametes placed on top of each row and each column. That's how you set it up. So then we're going to fill it in the same way we would fill in the box on a monohybrid. So the capital T and capital Y from this will go down. The little t and the little y will go across. Notice you put the t's together and the y's together. Put the capital first and the lowercase second. So we fill them in across and fill them in down. And really how you do it, do you want to fill in this box all at the same time, or fill in all the capital T's here, all the capital T's here, all the capital T's here, all the capital T's here? It's really a preference, but you want to make sure that you do it in the same pattern. Don't start changing how you're doing it in the middle, because you're more likely to make an error. So like I said, you put like terms together, put capital first. So this is your completed box. So this is what it would look like. So this is step number 3. So then what do? We need to read the information in the box. So if you notice, I have a genotype ratio of 16 out of 16, or 100% of them are going to be the heterozygous genotype. Capital T, capital Y, little y. So that means 100% of them will be yellow and will be tall. Okay, so they're yellow and tall, as opposed to yellow and short, or green and short, or green and tall. So 100% of them are going to have the yellow and tall combination. So this is how we fill out a dihybrid cross. So remember, it's the same 4 parts. You're going to identify the parent genotypes. You're going to determine the genotypes using that FOIL method. First letter with the second gene, first letter with the other gene. Go to the second one. Okay, remember that FOIL. You'll fill it in, making sure however you fill it in, you do it consistently. Do you go down each row, go across? Are you going to fill in each box? However you do it, be consistent. And then you're going to be reading the genotypes and phenotypes. So a little trick though on the genotypes and phenotypes, find one box and then look through. Do I see the same genotype of that first box? Okay, count them up. Okay, go to the next box. Do I see the same genotype as the second box anywhere else? Count them up. That's how I recommend doing it until you get to all of the boxes have been accounted for. If you need to just make a mark through your box that you counted it in one of your genotypes, do that. Make a mark, change the color. So you are going to do a little practice here on your own on your notes.